Glycolysis is a multi-step process that is used to convert glucose into 2-pyruvate molecules. To begin, glucose enters the cell using specific transport proteins. The glucose molecule is then phosphorylated to form glucose 6-phosphate with the help of ATP and the enzyme hexokinase. ATP provides the phosphate group for the phosphorylation of glucose, and the enzyme hexokinase catalyzes the transfer of the phosphoryl group from ATP to glucose. The result is glucose 6-phosphate molecule, a proton, and ADP.
The next step is the isomerization of glucose 6-phosphate to fructose 6-phosphate. In this step, the aldose is converted into a ketose. To start, the glucose 6-phosphate is broken open and isomerized. It is then closed to form fructose 6-phosphate.
To continue glycolysis, the fructose 6-phosphate is then phosphorylated into fructose 1,6-bisphosphate. Again, ATP provides a phosphate group. However, this time, phosphofructokinase is the enzyme used to catalyze the phosphorylation. The result is fructose 1,6-bisphosphate, a proton, and ADP.
Cleaving of the sugar is the next step in glycolysis. The fructose 1,6-bisphosphate is first opened, and then it is cleaved into dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, GAP.
GAP continues along the glycolytic pathway. DHAP, however, cannot continue on the pathway of glycolysis in its form, so it must first be converted into GAP. This is done through a reaction using triose phosphate isomerase. The reaction is in equilibrium, but the surrounding environment drives the reaction towards the production of GAP.
Now that there are 2 GAP molecules, they can be oxidized and phosphorylated. This is done through the addition of NAD+ and inorganic phosphate. Glyceraldehyde 3-phosphate dehydrogenase helps to drive the reaction. The result is 1,3-bisphosphoglycerate. For this reaction, the GAP molecules first undergo oxidation, resulting in the formation of a carboxylic acid. The carboxylic acid intermediate then undergoes dehydration, forming 1,3-bisphosphoglycerate.
ATP is formed from 1,3-bisphosphoglycerate. This is done through use of the enzyme phosphoglycerate kinase. It dephosphorylates the 1,3-bisphosphoglycerate molecules and attaches the phosphate to ADP, resulting in the formation of ATP and 3-phosphoglycerate.
Next, the 3-phosphoglycerate molecules are converted to 2-phosphoglycerate with the phosphoglycerate mutase to rearrange the molecules of 3-phosphoglycerate.
Then, for the final step, the 2-phosphoglycerate molecules are converted to pyruvate. First, enolase converts 2-phosphoglycerate to phosphenol pyruvate intermediate. From there, the enzyme pyruvate kinase helps to continue the reaction to form pyruvate.
Because 2 GAP molecules follow along the glycolytic pathway following cleavage of the sugar, the result is 2 pyruvate molecules and 2 ATP molecules.
Overall, glycolysis is a biological pathway that converts glucose into pyruvate and usable cellular energy, ATP, making it an essential pathway for organisms to create and regulate energy.